Thank you for the introduction. I would like to express my utmost gratitude to the chairman of the board, Dr. Yazawa, for giving me this opportunity. I would like to make a presentation under this title. As a chemist, I focus mainly on researches from the aspect of chemistry. As mentioned by Dr. Shirakawa earlier, there are two types of naturally occurring vitamin K. Vitamin K1, which is found in abundance in these green and yellow leafy vegetables, and vitamin K2, which is found in great amount in fermented foods. In particular, as vitamin K2, menaquinone N is named differently depending on the number of repetition of its side chain. They are also known in the contracted form, MKN. Among them, menaquinone 4, which has 4 repeating units as its side chain structure, is the body's most active vitamin K, and is found plenty in meat and eggs. On the other hand, vitamin K3, which has no side chain structure, is known to be a metabolite of vitamin K. The most basic physiological function of vitamin K known is its gamma carboxylation reaction. Through this basic metabolic cycle, which is called vitamin K cycle, vitamin K acts as a coenzyme of gamma-glutamyl carboxylase and leads to the conversion of vitamin K-dependent protein into its activated form, which is exhibited as blood coagulation and bone formation. Other than these, recently a variety of physiological functions of vitamin K have been discovered, one being the transcription regulation function of menaquinone 4 through the nuclear receptor SXR. We found that firstly MK4 binds to the SXR to form heterodimer, and the heterodimer then binds to the response element, causing gene expression and inducing the expression of metabolic enzyme CYP3A4 and factors such as JUXI and matrilin-2, which are both involved in bone formation. Additionally, vitamin K has also been shown to have other effects related to the brain. In the brain, differentiation of neurostem cells into neurons, astrocytes, and oligodendrocytes is known to be responsible for the higher functions of the brain. Very recently, our group discovered that vitamin K can selectively differentiate neurostem cells into neurons. Although presently, the mechanism behind is still unclear, it is believed that there is a possibility that vitamin K's physiological function can be applied to compound in the restoration of brain functions. In this way, we were able to find a large variety of activities of vitamin K. Particularly, we focused on the effects of vitamin K on SXR and the induction of neuronal differentiation of SXR. We aim to synthesize compounds with enhanced biological activities through derivatization. Firstly, let's talk about the investigation done on novel vitamin K derivatives with agonistic effect on SXR. As mentioned earlier, MK4 has been shown to exert its effect on transcription regulation through the nuclear receptor SXR. As shown on the left-hand side of this slide, SXR has a 3-dimensional structure, and this area is known to be a ligand-binding site. On the right side, examples of compounds with high SXR agonistic activities, such as rifampicin and hyperferon, are shown. Other examples include lipocholic acid, and as mentioned earlier, naturally occurring MK4. Based on the structures of menaquinone-like compound, for example, MK4, vitamin K derivatives with potent agonist activity were designed and chemically synthesized through efforts in chemical modification. Here, the evaluation methods for SXR-mediated transcriptional activity are described. Two types of luciferase assay methods are used. The first one is a luciferase assay that utilizes a CYP3A4 promoter containing SXRE. When the ligand binds to SXR, RXR will bind to the ligand SXR complex to form a heterodimer. The binding of the heterodimer to SXRE serves as the basis for the analysis of the transcriptional activity. The other method is a one-hybrid luciferase assay that utilizes SXR-GLA4 complex instead. SXR firstly binds to GLA4, and the ligand then binds to SXR part of the SXR-GLA4 complex. This is the method where the direct effect of the ligand on transcription activity is analyzed. Using the two above methods, vitamin K's SXR-mediated transcriptional activity can be evaluated. Firstly, the SXR-mediated transcriptional activities of naturally occurring menaquinone-type compounds were evaluated. Rifampicin was used as a positive control during the evaluation. We measured the transcriptional activity of menaquinone compounds with side chain of different number of repeating units, from MK0 to MK7, where MK0 represents menadione. Although the activities were not as much as that of rifampicin, MK2 to MK4 were found to have high activities. With these results, we decided to perform chemical modifications to the side chains of MK2 to MK4 when experimenting. Due to the high activities of menaquinone compounds, MK2, 3, and 4, shown in the previous assays, we investigated the changes to the transcription activities of menaquinone compounds when these menaquinone derivatives with two equal side chains of MK2, 3, and 4 respectively are synthesized. The results of the investigation with synthesized vitamin K derivatives are shown here. The new vitamin K derivatives are named M, K, and W, and the transcription activities were evaluated using the two previously mentioned methods. It is found that MK2W, which has two side chains introduced, exhibited the highest activity. The order of activities of intensity are shown here. On the other hand, among naturally occurring vitamin K, MK3 was found to have the highest activity. In other words, the transcriptional inducing activity of vitamin K derivative is significantly influenced by the length and size of its side chains. Next, we decided to investigate the transcriptional activity of vitamin K derivatives with modified side chain parts. Natural menaquinone compounds with a hydroxyl group as the most hydrophilic side chain incorporated at the end, and contrarily, those with a benzene ring or phenyl group added to the same position were synthesized, and the transcription activities were examined. These are the results of the experiment. Compared to the naturally occurring menaquinones, MK1 to MK4, the activities of MK1 to MK4 with a hydroxyl group added to the end of the side chain were found to be reduced compared to the natural counterparts, whereas the transcriptional activities of MK2 and MK3 with a benzene ring added to the end of the side chain respectively were seen to be significantly increased. In particular, it was revealed that the menaquinone compound in which a benzene ring is introduced at the end of MK3 showed an equal or higher transcriptional activity than that of the positive control rifampicin. Therefore, the introduction of a benzene ring at the end of a side chain of vitamin K can produce a derivative with strong agonistic activity. In order to understand the mechanism behind the binding of these previously mentioned modified derivatives, those with two side chains and those with benzene added to the side chain of the compound, a docking study using in silico analysis to analyze binding SXR was performed. This is a docking study where the interaction between the two most active compounds, MK2W and MK3 with a benzene ring added to the end, and SXR are investigated. The results show that the hydrophobic interaction between the lipophilic part of the ligand compound and the lipophilic amino acid that is part of the SXR is important. In other words, this analysis shows that a moderate degree of hydrophobicity is necessary for the agonist activity of this ligand. To conclude, we were able to demonstrate the structural specificity of vitamin K for the transcriptional activity of SXR using this experiment. Moving on, it is about the investigation of novel vitamin K derivatives with the ability to induce differentiation of brain neural progenitor cells into neurons. As mentioned earlier, the differentiation of neural progenitor cells into neural precursor cells and glial precursor cells, and further differentiation into neurons, astrocytes, and oligodendrocytes are known to be responsible for the higher functions of the brain. Very recently, our group has shown that some forms of vitamin K can induce the differentiation of neural progenitor cells into neurons. This effect has been shown to vary in intensity depending on the structure of the side chain of vitamin K. We believe that if we can find compounds with high differentiation-inducing activity, we might be able to make use of them in the recovery of brain function. Thereby, we synthesized compounds with various changes to the side chain of vitamin K, all with the aim of creating compounds with high differentiation-inducing activity. When we were thinking about the way to structurally modify vitamin K, we took into account that the brain is composed of mainly fat-soluble substances, and hence designed and synthesized vitamin Ks with various fat-soluble substituents, primarily compounds with benzene rings introduced at the end of the side chain of vitamin K. We believed by doing so, the differentiation-inducing activity can be increased. The top row shows a side chain that are primarily structures with benzene ring, such as naphthalene or biophenol. The middle row are side chains that are basically benzene ring with various substituents. And the bottom table summarizes those with addition of methyl groups at various positions. In all of them, the vitamin K part is either MK2 or MK3. In this slide, the evaluation methods for the differentiation-inducing activities of vitamin K derivatives are shown. Firstly, neural stem cells were extracted from the cerebrum of embryonic mice of the age of 14 to 19-day-old. The stem cells were then seeded and cultured for 24 hours before a variety of vitamin K derivatives were added at a concentration of 1 micromolar, and then cultured for 4 days. The differentiated neurons were later stained using immunofluorescence staining, where neurons were stained red and astrocytes were stained green. Finally, the results were visualized under confocal laser microscope or fluorescence microscope. And this is how the differentiation looked. The results are shown here. The FNO here indicates that no vitamin K was added. As you can see, the red area, which represents differentiated neurons, are almost completely absent. In contrast, here are the results when 1 micromolar of phyloquinone, also known as vitamin K1, and 1 micromolar of MK4 is added respectively. Since all trans-retinoic acid is known to be a substance with high differentiation-inducing activity, it was used as a positive control. The results show that among vitamin K, stem cells administered with MK4 produced more red areas and therefore more differentiated neurons, suggesting a high transcription-inducing activity. This is a summary of results studying the activities of the naturally occurring MK4 and other synthetic compounds. As I've mentioned earlier, the administration of MK4 to the FNO group can result in significant promotion of differentiation. The synthetic compound MK3-MMP, which contains the meta-methylphenol group at the end of the side chain, is even more potent. この MK3 の末端にメタメチルフェニル基を導入した MK3-MMP と名付けた化合物は、さらに分化を促進するということが明らかになりました。In other words, this compound is more potent than MK4 in inducing differentiation. On the contrary, the compound with the benzene ring MK2-DMP, which has 2 methyl groups added to the side chain of MK2, has a much less area in red and has the opposite effect in differentiation. These results suggest that vitamin K compounds may be able to control neural differentiation. The mechanism of action is not yet clear, but elucidation of this mechanism may be useful in the development of compounds to enhance or suppress these activities. In order to elucidate the mechanism of action and unravel the ways in which these compounds and the target proteins interact, we decided to synthesize compounds, including labeled compounds, starting with biotin-labeled vitamin K and various derivatives of vitamin K. Two possible ways can be used to isolate and identify the proteins to which vitamin K binds. One is the pull-down method, in which the vitamin K binding protein is specifically fished out and identified in this way. And the other is a fluorescent method, in which the vitamin K binding protein is made to glow. First of all, let me explain the pull-down method. First, we have to label this compound with biotin. As the side chain is important for activity, they must be avoided when labeling. Contrarily, the furthest part from the side chain is labeled instead. In other words, a compound in which biotin is introduced via linker at position 7, the furthest position from the naphthaquinone ring is emphasized. This compound is then used to fish out the protein. In this case, we use the pull-down method to specifically capture proteins that are bound to the menaquinones. To perform this process, firstly vitamin K compounds are labeled with biotin. On one hand, porcelain beads, which are polymers that are stuck to magnets, are attached with avidin. In this way, the porcelain with the avidin beads become bound to the biotin-labeled vitamin K. This biotin-bound vitamin K acts as a hook to fish out specific binding protein. In other words, by introducing biotin-labeled vitamin K-bound porcelain beads to a cell lysate prepared from crushed neural stem cells and shaking it, the desired proteins will recognize the vitamin K attached and bind specifically to it. Lastly, while holding the porcelain beads in place with a magnet, the unwanted protein is then washed out, isolating specific proteins in the process. This shows the bands of results of a preliminary purification experiment using STA's page to further isolate the MK4-bound protein isolated from HepG2 cells using the previously mentioned pull-down method. Further analysis of band A using MALDI-TOF-MS revealed this to be a previously reported MK4 binding protein, 17-beta-HSD4. This indicates the possibility to practically use the mentioned method to isolate MK4 bound proteins. Presently, neural stem cells are being used to analyze this binding protein. Although I'm not able to present our result this time, I can tell you that we were able to isolate and identify proteins related to neural differentiation. The other method I would like to share with you is the identification of vitamin K-bound proteins using fluorescent labeling. In this protein fluorescent labeling method, NBD, 4-nitrobenzyl-2-oxa-1,3-diazo, a type of fluorescent material is chosen. This fluorophore is unique in a way that it does not exhibit fluorescence when oxygen or halogen is attached to this part, X, but it does so when nitrogen is attached to it. When NBD is bound to other compounds via bond formation through its oxygen atom, no fluorescence is observed. However, when the fluorescently labeled compound is bound to a protein, if there happens to be an amino group of a basic amino acid within the vicinity of the protein, NBD will be attacked by the protein. In other words, a bond will be formed between the nitrogen atom and the fluorescent compound, transferring the fluorescent group to the protein in the process. As a result, the protein is now fluorescently labeled. This method was used to label vitamin K. This is an NBD fluorescent labeling material with MK2 bound to the ligand part. In this slide, it is seen that fluorescence occurs when bond formation through nitrogen atom occurs. But if the bond is formed with oxygen atom, no fluorescence is observed. The respective intermediates are named NBD2 and ONBD2 respectively. In the case of NNBD2, the fluorescence is present from the start, which means that it is always fluorescing even when it is not bound to any protein. On the other hand, if NNBD2 is bound to oxygen atom, it becomes non-autofluorescent. But similarly to when NBD2 is bound to a protein, the basic amino acids present in the protein will attack the NBD in this way, and the fluorescent part of NBD will be transferred to the protein via bond formation with the nitrogen atom. In this way, the binding protein becomes fluorescently labeled. The first step was to verify whether the compound exhibits autofluorescence. To do so, the autofluorescence of the compounds was confirmed using a fluorescence microscopy. The results show that MK2 itself does not exhibit autofluorescence. On the other hand, NNB2 does fluoresce when it forms a bond with a nitrogen atom. And ONBD2 does not autofluoresce at all, as shown here. The next step was to check if this non-autofluorescent ONBD2 could indeed be used to fluorescently label vitamin K-bound proteins.
結合タンパク質を経口標識できるのかを確認しました。これは ONBD2
In this experiment, ONBD2 was added to MG63 cells, which are SXR nuclear receptor overexpressed bone
先ほど
cells.
ご紹介した SXR、これビタミン K が結合する核内受容体ですが、
Upon adding ONBD2 to the cells, fluorescence of NBD was observed. This indicates that ONBD2 have bound to SXR, and it is fluorescently labeled by NBD.
NBD の傾向が見られました。これは SXR に ONBD が結合して、蛍光物質、それによって、NBD 標識、NBD の傾向が蛍光標識されたことを示しています。
Continuous observation over time helped us understand that the fluorescence of the NBDs behave in the same way as SXR, confirming that ONBD2 can be used to fluorescently label vitamin K bound proteins.
このビタミン K の結合タンパク質を蛍光標識できるということが確認できました。
Currently, neural stem cells are used in our analysis.
解析を行っております。
This is a summary of the items mentioned above. We have demonstrated that it is possible to enhance various activities of vitamin K by derivatization. Moreover, it is possible to isolate and identify vitamin K-bound proteins using the pull-down method and the fluorescent labeling method. With this knowledge, we will develop more highly active compounds in the future. This is the end of the presentation part, but I would also like to express my sincere gratitude to all those who have helped us in our researches. Furthermore, we are very grateful to be supported by research grants such as this one. Thank you very much for your attention. This is the end of the presentation.